and welcome to A Perspective on Crime, a podcast that explores contemporary research in criminology. Normally, with each episode, I prattle on for a couple of minutes with some self-regarding nonsense with reference to the research we're going to explore. Is it egotistical? Definitely. The research we're covering today, though, needs no embellishment, no hyping, no warm-up act. It's about an aspect of the world that we all adore. Dogs. The paper is titled Investigation of Human DNA Transfer During a Mock Dog Napping and was written by lead author Heidi Monkman and her colleagues and was published in the journal Forensic Science International. It examines the scenario of a dog napping and asks whether dogs could also be a forensic investigator's best friend, a living, breathing, furry host and courier of the napper's DNA. It's a piece of work that forces us to look at our four-legged companions in an entirely new light, and it poses a central question. Just how effectively can a dog act as a vector, picking up DNA from one person and depositing it in a completely different location? The answer, as we'll see, could have profound implications for how crimes are investigated in the future. But let's start from the first principle. Pet theft, and particularly dog theft, is a deeply distressing and increasingly common crime. It's an issue that causes immense emotional damage, and it's a global one. To put it in perspective, in the UK an estimated 5 pets are stolen every single day. Each of these incidents represents a family torn apart, and this issue the researchers in Australia at Flinders University sought to address. The challenge is that prosecuting these cases is notoriously difficult. The clandestine nature of the act, combined with the scarcity of physical evidence, often leaves law enforcement with very few leads, pardon the pun. How do you link a suspect to a stolen animal when the crime happens in seconds and leaves no obvious trace? This is precisely the evidential gap that Heidi Monkman and her team set out to explore. They wanted to see if the dog itself could be the key, carrying a thief's DNA and transferring it to other locations, like a getaway vehicle. To do this, they designed a brilliantly simple yet rigorously controlled experiment, a mock dog napping. The experiment involved several key players, all carefully chosen to avoid any pre-existing contamination. First, there were 5 different dogs, a wonderful variety of breeds, ranging from a Cavadoodle and a Maltese Shih Tzu cross to a Border Collie, a Doberman cross and a Greyhound. These weren't just test subjects, they were family pets, participating with their owner's consent. Then there were 5 different cars, each one completely unfamiliar to the dogs and their owners. This is a critical detail. No prior contact means no pre-existing DNA. And finally, there was a single handler, a person experienced with animals who would play the role of the mock dog napper for each of the 5 experiments. Control here is absolutely critical. Think of it like setting a perfectly clean stage before the play begins. The dog owners had never been in the cars. The car owners had never met the dogs, and the handler had no prior contact with either the dogs, their owners, or, in 4 out of the 5 cases, the cars. This clean slate ensured that any DNA found later could be traced back directly and with confidence to the events of the experiment itself. The procedure, as detailed in the study, followed a clear, logical sequence, almost like a short scripted scene. For each of the 5 experiments, which were conducted on separate days to prevent any carryover, the handler would arrive at the dog's home. After a brief and gentle introduction, just a few pats to get acquainted, the handler would lift the dog and carry it to the waiting car, a short distance away. The researcher, clad in full personal protective equipment to avoid adding their own DNA to the mix, would open the car door. The handler would then carefully place the dog onto the back seat. For the next 20 minutes, a clock was ticking. The handler sat in the driver's seat and the researcher sat in the passenger seat, not to drive, but to monitor the dog's welfare and observe its movements. After those 20 minutes were up, the process was reversed. The handler carefully lifted the dog out of the car and carried it back to its doorstep. The entire interaction was designed to realistically simulate a quick, opportunistic theft. But the real scientific work, of course, began after the crime was completed. This is when the forensic investigation truly started. The researchers began the meticulous process of sampling for DNA. 1 hour after being returned home, each dog was swabbed on its head, back and sides. 4 hours after the experiment, various surfaces inside the car, locations the researchers observed the dog touching, were sampled. And finally, the shirt worn by the handler was collected for analysis. In a clever touch to simulate real-world evidence discovery, the handler wore the shirt for several hours after the event, then placed it on the bedroom floor overnight, mimicking how an item of clothing might be found in a suspect's residence. The next day, specific areas of the shirt that had contacted with the dog, the arms, abdomen and chest, were also sampled. This comprehensive collection strategy created a forensic map, a way for scientists to follow the invisible trail of clues left behind during that brief 20-minute mock dog napping. With all the samples collected and carefully catalogued, the researchers at Flinders University set about answering the critical investigative question at the heart of their study. They were looking for a trail of invisible clues, a microscopic story told through DNA. What they found was both fascinating and, for the world of forensics, deeply significant. First, as you might expect, the dog owner's DNA was everywhere on their own pets. It was detected in a remarkable 85% of all samples taken from the dog's coats. This wasn't a surprise, in fact, it was an important baseline. It confirmed what other studies had shown. Our dogs are walking reservoirs of our own genetic material. But the truly critical finding came when they looked for the handler's DNA, the DNA of the mock thief. They found the handler's DNA on 40% of the dog cases. This is a significant result. It means that even from a brief period of being carried, there was nearly 2 in 1 chance that the perpetrator's DNA would be left on the animal. But the researchers dug deeper and noticed an interesting pattern. The handler's DNA was only found on the 2 largest dogs in the study, the Doberman Cross and the Greyhound. The sources provide a clear explanation for this. The contact time with these larger dogs was longer, around 2 minutes of being held, compared to the much shorter 15 and 37 seconds for the last 3 smaller dogs. The larger, heavier dogs required more physical contact to carry securely, with the handler's arms and shirt pressing against the centre and belly of the dog. This suggests a direct link. More time and more surface area contact equals a higher likelihood of DNA transfer. It's a textbook example of Locard's exchange principle in action, every contact leaving a trace. Next, the investigation turned to the car, the secondary crime scene. This was where the concept of the dog as a vector of transfer really comes into play. The researchers looked for the dog's owner's DNA inside the vehicle, and they found it in 35% of all samples. Let's pause and consider that for a second. The dog owner was never in or even near the car. The DNA could only have gotten there one way. It was carried from the home on the dog's fur and then shared inside the car during that 20-minute period. This is what forensic scientists called indirect transfer, and it demonstrates powerfully that a dog can act as a living bridge, moving evidence from one environment to another. But it wasn't just the owner's DNA being moved. In a fascinating twist, the handler's DNA was also found in the car, specifically in 13% of the relevant samples. Since the handler only sat in the front seat and never touched the back where the sample was taken from, their DNA was most likely transferred by the dog. The dog picked it up while being carried and then deposited into the car's back seat. This multi-step transfer from handler to dog to car is exactly the kind of evidence that could link a suspect to a vehicle used in a crime. But the chain of transfer didn't stop there. The researchers also detected the car owner's DNA in 5% of the samples taken from the dog. The car owner had never met the dogs. Their DNA, presumably shared onto the car seats over time, was picked up by the dogs during their 20-minute stay and was still detectable on the fur an hour later. Finally, the scientists examined the handler's shirt. Here, they were looking for evidence transferred in the opposite direction, and they found it. The dog owner's DNA was detected in 10% of the samples taken from the handler's shirt. This is a classic example of secondary transfer. The owner's DNA was on the dog's fur, and when the handler picked up the dog, that DNA was transferred from the dog onto their clothing. Even more striking, the car owner's DNA, from the car seat to the dog to the handler, was also found on the handler's shirt in 5% of samples. This could be incredibly valuable in an investigation, creating a potential link between the suspect's clothing and both the owner of the stolen pet and the vehicle used in the crime. It's a nuance that highlights the complexities in this field. The study noted that the handler's shirt with the most of their own DNA on it did not lead to a detectable transfer from the dogs, while the shirt with the least DNA did. This suggests that the starting amount of DNA isn't the only factor. The dynamics of transfer are unpredictable and require careful study. Across all of these samples, from the dogs, the cars and the shirts, the researchers frequently detected unknown DNA profiles belonging to neither the owners, the handler, nor the car users, highlighting the reality that our world is saturated with DNA. We shed it constantly, and it gets moved around through all sorts of casual, everyday contact. The presence of this background DNA doesn't invalidate other findings. Rather, it underscores the complexity of interpreting trace evidence. In fact, as we'll discuss later, this background noise is precisely why studies like this one are so vital. What this research provides, in summary, is a clear proof of concept. In a simulated crime, a dog can and does act as a dynamic participant in the exchange of DNA, picking it up and dropping it off in a way that creates a traceable, albeit invisible, evidentiary trail. The implications of these findings stretch far beyond the laboratory. For law enforcement and forensic investigators, this research provides a powerful argument for a new and fundamentally simple step in evidence collection that is very often overlooked. It suggests that at any crime scene involving a pet, be it a theft, an instance of abuse, or a situation where a pet has gone missing and is later recovered, the animal itself should be considered a primary source of evidence. The current protocols might focus on dusting for fingerprints on a broken window or swabbing a door handle, but this study argues that investigation should also be carefully, methodically swabbing the animal's coat for the perpetrator's DNA. The research effectively reframes our understanding of the role an animal can play in an investigation. It asks us not to think of a dog just as a victim or a piece of property, but as a potential DNA vector. A simple but effective way to visualise this is to think of the dog as a kind of furry courier, a living evidence bag with 4 legs. Throughout its day, it passively connects a baseline of DNA from its owners and its home environment, all over its fur. If a stranger like a thief then makes physical contact, the dog picks up their DNA as well, adding it to its existing collection. When that thief moves the dog to a new location, like a car, the dog begins to act as a distributor, shredding this collected DNA, both from its owners and from the thief, into this new environment. The process creates a complex but potentially invaluable tapestry of evidence. That tapestry can link a suspect to an animal's owner to a suspect's car, and a suspect's clothing back to the animal's owner. The dog, in this sense, becomes the silent, central hub of a network of forensic connections. By linking these forensic possibilities directly back to the very real problem of dog theft in Britain and around the world, the research becomes more than just an academic exercise. It offers a tangible, scientific tool that could help law enforcement investigate the thousands of pet thefts that occur every year. For the police responding to the report of a stolen dog that's been found abandoned in a park, this study provides a clear, evidence-based rationale for treating the dog as a priority for forensic examination. The DNA recovered could be the key to identifying a suspect and securing conviction in a type of crime that has traditionally been very difficult to prosecute. This moves the conversation from the practical what of the findings, the DNA that's transferred, to the more complex and crucial so what of their application in the courtroom. It's not just about finding DNA, it's about being able to explain what its presence truly means. This brings us to one of the most sophisticated aspects of modern forensic science, and one where this study makes a crucial contribution. In today's courtrooms, finding a suspect's DNA at the crime scene is often no longer the slam dunk it was. The defence will rightly ask, OK, so my client's DNA is there, but how did it get there? And when? Was it from direct involvement in the crime, or could it have arrived through some innocent, indirect route? Answering this question is known as active level evaluation, and it's the modern frontier of DNA interpretation. It's about moving beyond identifying whose DNA it is and towards evaluating the likelihood of different scenarios that could explain its presence. Remember all of that unknown, background DNA we talked about earlier. That's precisely why activity level evaluation is so critical. The world is full of innocent DNA transfer, so the challenge is to provide the court with a scientific way of distinguishing between the DNA that's relevant to the crime and all the other incidental DNA that just happens to be there. This study provides exactly that kind of empirical data that forensic scientists need to perform these crucial evaluations. To do this, the researcher uses a powerful statistical tool called the Bayesian network, or BN. Now, this might sound some intimidating and technical term, but we can think of it more simply as a kind of probability engine. It's a computer model that allows forensic scientists to input data from studies like this one and weigh competing scenarios against each other to see which one is more probable, given the DNA evidence found. The paper provides a perfect, real-world example. Imagine a person of interest is accused of stealing a dog. Let's consider two competing propositions, one from the prosecution and one from the defence. Scenario 1, the prosecution's view. It is that the person of interest is the one who actually held and carried the dog during the theft. Scenario 2, the defence's view, is that an unknown alternate offender carried the dog and the person of interest DNA was transferred indirectly. Perhaps the defence might argue that the person of interest lives with a friend who recently visited the dog's home and patted the dog, innocently transferring his DNA from their shared environment onto the dog's fur. Without data, a court's left to speculate, but with the data from this mock dog napping study, the Bayesian network can start to calculate probabilities. The model knows, based on research, how much DNA is typically transferred when someone carries a dog. It also knows from other studies how much DNA might be transferred from a casual pet. By comparing the amount of the person of interest DNA found on the dog in the actual case to the amounts found in these different experimental scenarios, the network can calculate what's known as a likelihood ratio. This ratio tells the court how much more likely the DNA evidence is if its prosecutor's scenario is true, compared to if the defence scenarios is true. It doesn't prove innocence or guilt, but it gives the jury a scientific, data-driven way to assess the weight of the DNA evidence, which is far more powerful than relying on pure intuition or speculation. Of course, to maintain scientific credibility, It's essential to acknowledge the boundaries of any research, and the authors of this paper are transparent about the study's limitations. They highlight that their experiment involved a relatively small sample size, just 5 dogs, and focused on one very specific scenario, a brief dog napping where the dog was placed in the back seat of a car. This means the results, while incredibly valuable, serve as a foundational proof of concept rather than a definitive, universal rulebook for all dog-related DNA transfer. It's the first step on a longer road. Recognising these limits naturally points the way towards future research. The authors themselves propose several important next steps. They call for larger studies involving more dogs of different breeds and sizes to see if these initial patterns hold true on a bigger scale. They also suggest exploring different variables and scenarios. For instance, what happens if the dog's handled differently? Perhaps transported in the boot of the car instead of the back seat? What if there's a longer time frame between the criminal event and the collection of samples? How does that affect the persistence of the transferred DNA? There's even the exciting potential to incorporate the dog's own DNA as evidence. Using newly developed canine DNA assays, it might be possible to link a specific dog to a suspect's car or home, adding another layer of forensic information to an investigation. This pilot study, despite its limitations, has answered an initial question while simultaneously opened up a whole new field of inquiry, providing a vital framework for future work that will build upon its findings. It seems our pets, and specifically our dogs, are far more than just companions. They're living, breathing archives of forensic information. They silently and continuously document the world around them through their invisible traces of DNA that they collect and carry on their fur. The study urges us to see the family dog not just as a potential victim in a crime like theft, but as a key witness whose testimony is written in a genetic code. Evidence can be found in the most unexpected and familiar of places. For any listeners who wish to delve into the details themselves, the research paper we've discussed today is titled An Investigation of Human DNA Transfer During a Mock Dog Napping. It's authored by Heidi Monkman and the research team, and you can find it published in the journal Forensic Science International. A link to the paper, as ever, is in the show notes, and of course, I'd urge you to read it for yourself. New episodes will drop every week, so make sure you subscribe, and please recommend A Perspective on Crime to friends, colleagues, and anyone you think might be interested. Finally, if you've read some research that you think others should know about, drop me a message via the link in the show notes. And if you yourself have new work coming out and like it covered, please get in touch. Until next time, goodbye.